so excited to see how things have gone. With no further ado, we'll get to that. And then if you have to, if people need to run, you need to run. I'll try to send out the week 4 survey. We may try to do a small discussion after we're done about how this course could be improved, what went well, what didn't. We'll try to make that as painless as possible and get everyone out in decent time to enjoy a weekend that actually looks nice. And yes.
Yeah. Also, we will have the Discord active for a while because other sites are still going and are still running. So feel free to join the pause if you have interesting topics and to join the conversation, interact with each other, and don't forget to submit your projects.
Yes, and those for those taking through the IMS course, don't forget to submit your projects. And for those not taking through the IMS course, think about submitting your projects because it's kind of cool too. It's a cool way to incorporate your project status or your project updates into like BrainHack going forward.
And send us your slides as well on just upload it to the Google folder. So for whoever's presenting, just share your screen and then unmute yourself so we could record your presentation. Okay.
If you put the text or I don't want to. Yeah, let's just. Are you figuring to kick to? Yeah, I'll click. I feel like I like natural. I don't know. That's right. So that's usually supposed to be muted. Yes, green should be. You have to like. Oh. I'm sorry. Usually they glow.
It doesn't have a mute button on the bottom left anymore.
Oh.
Yeah, I can hear you now, but it's not very clear. We might want to put one in front of them or something.
Hello.
Oh.
Here.
Yeah.
You're here?
The quality is very good though.
Oh.
Just try to speak up.
Okay, we can project. Do that. We can try. We good to get started? Yes, this is where I'm gonna stand over here.
Okay, sure. Okay. Okay, good afternoon, everyone. We're really happy to present our project that we've been working on for this past month. I'm just gonna reintroduce all of us here. So I'm Kayla. We have Anjali, we have Therese, we have Janine, and Leanne, and we also had Quinn. She's just not here today, but we're really excited to present our project on fMRI signatures and behavioral correlates of self and empathic pain. So first, I just want to get us a little bit oriented to some of these concepts. So empathy, as you are probably already aware, is an individual's ability to perceive, understand, and respond appropriately to others' feelings as well as behavior. But specifically empathic pain is describing individuals' perception, judgment, and or emotional response to someone else's pain or suffering. And extant literature shows that it's a multidimensional, complex social cognitive ability that is involved in a lot of very healthy interactions between people and also daily functioning. However, their underlying neurobiological mechanisms that support empathic pain are not fully characterized. So, background literature and convergent evidence shows that both human and animal studies have multiple brain regions that are implicated in empathic pain. So these are including, but not limited to, the insula cortex, the anterior cingulate cortex, the mid-cingulate cortex, and nucleus accumbens, as well as the inferior frontal gyrus, medial frontal gyrus, and postcentral gyrus. And we also don't know how these might be related to other aspects of behavior like loneliness or social connectedness. So we have several research questions that we aim to assess in our project. First, what regions are activated during the self-experienced pain versus empathic pain tasks? Does loneliness or social connectedness at baseline predict activation in the brain during self-experienced pain versus empathic pain tasks? As well as functional connectivity. So are there any differences in functional connectivity between these two conditions? And is functional connectivity even associated with loneliness or social connectedness? So we had a few objectives throughout our project. First, how do we analyze fMRI data? This is very, very new to us. We did not do this in the past, so there was a lot to learn in both analyzing activation as well as connectivity in a task-based fMRI, and how to visualize our outcomes. We had to learn how to use Python and GitHub and create shared repositories, which was its own task. And then we also wanted to earn course credit in the meantime. So I'm going to hand off to Lee now just to describe our data set.
So the data set we used was the empathic pain task, which is an open source data on OpenNeuro. It included task-based fMRI, like we said. The data was pre-processed using fMRI prep, so we didn't do that part of it. And they were T2 star scans, repetition time was 2,500 milliseconds. And the data set actually also included behavioral data, which was very cool. So it included data on loneliness and social connectedness. There were 54 participants involved in this study. They were grouped into two different meditation groups. The first group is the loving kindness meditation group. There were 25 participants in that one. And then the progressive muscle relaxation group served like a control group. And those were 29 participants. The specific task that we wanted to analyze has two runs. There's a self-experienced pain and an observed pain portion. So in the observed pain portion right here, like the one on the top, basically participants get an auditory cue, and then they basically see a study partner, that's what they called them, getting like a pain cue or something. And that lasted for around 6 seconds, and then they go back to rest. In the self-experienced pain run, what happened is they also listened to an auditory cue. And then after that, they themselves received the pain stimulus for again, around 6 seconds, and then rest. So there was a rest period after that. So for each run, there were 30 trials, 24 seconds each. The total acquisition time was 750 seconds, but the task specifically was 720 seconds. Now, the tools that we use to analyze this data, so software, we use Python, MS Visual Code, Studio Code, Nylearn, and R Studio. We use GitHub, different Python scripts and Jupyter notebooks for our codes and project management. And then finally, for our computing environment, just use our local laptops. And I'm going to hand it off to Anjali to talk about the specific analysis we did for task activation.
Yeah, so I'm just going to start off by kind of providing an overview of how we conducted the analysis, but specifically for the task activation portion. And then once I'm finished, we'll discuss the functional connectivity analysis in a bit more detail. So regarding the preliminary part of the analysis, I've kind of broken it up into 4 main parts. First, it'd be data preparation, followed by the events file parsing, the first level generalized linear model that we ran, and then finally cluster detection. Then I'll move on to the secondary part after. So regarding data preparation, we first loaded the pre-processed bold fMRI data for each subject. We then trimmed the first and last time points to remove edge artifacts as instructed by the actual researchers who collected this data. We also loaded the confound regressors and ensured that they were aligned with the bold time points. Regarding the event file parsing, for each trial type, we loaded the event timing files, specifying the onset and duration for each file. We also constructed any events data frame for each task. For the first level GLM that we ran, we fitted it using Nylearn's first level model, modeling the bold response to each task. We were sure to include the confound regressors in the model as well. And then we computed Z-maps for each task construct, contrast in relation to the baseline. Regarding the cluster detection, we applied a statistical threshold of greater than 3.1 to each Z-map. We then identified significant clusters using Nylearn's get clusters table. And then finally, we labeled the cluster peaks using the Harvard-Oxford Atlas. Should also be mentioned that we did try to apply a threshold correction, but unfortunately, none of our signals were really strong enough to survive that. So regarding the task activation analysis, it is not correct using FDR or FWE correction. So now regarding the secondary part of this analysis, we then summarize the activation for each region of interest using the mean peak statistic for every subject and task. And then after this, to answer kind of our two research questions regarding task activation, we first generated a heat map looking at the ROI activation for each task. We then ran a MANOVA looking at the interaction of the different group conditions and the behavioral measures that we were interested in, looking at whether there was a relation between that and activation during the self-pain and empathic task specifically. And then finally, we decided to visualize this interaction using scatter plots generated in RStudio.
And for the functional connectivity part, for comparing the differences in functional connectivity between two conditions, so we first, for each condition, we apply the Harvard-Oxford Atlas, which contains like 69 parcellations, and we applied a mascot. And then we applied Pearson correlation and generated a 69 times 69 matrix per condition per subject. And we then tried to visualize it based on like the different networks, which we first reordered these ROIs based on like different networks, for example, the salience network or DMN, and we generated the connectivity matrix figures. And we also like highlighted the network boundaries for like better visualization. And for the statistical analysis part, we flattened the upper triangle for each subject's matrix, excluding the diagonal line. And then we performed the edge-wise comparison using like paired t-tests.
So one of the other questions for the functional connectivity analysis, so other than checking whether or not there are differences in functional connectivity across the two run types, we're also interested in seeing whether functional connectivity was associated with behavioral measures such as loneliness or social connectedness. So after we obtained each participant's individual functional connectivity matrices, so we had both run 1 and run 2 connectivity matrices for each participant, then we separated these into like two groups. So we had the run type 1, so self, versus run type 2, empathic pain. And we're also interested only in ROI pairs that were implicated in self-pain and empathetic pain. So this included ROI pairs that had either the insula or the cingulate cortex as one of the ROIs in the pair. And then finally, for the statistical part of it, we ran linear regression models. So we wanted to see whether or not functional connectivity between ROI to ROI pairs was associated with loneliness or social connectedness. And we also included covariates such as age, sex, and condition.
So in terms of the task activation findings, they were a little bit disappointing. So our first question, again, was what regions are activated during the self-experience pain versus empathic pain tasks? And this was the heat map that we generated. So for the most part, activations look very similar between the empathic pain task and the self-pain task. regions that we wanted to focus on were the anterior cingulate cortex, insula, and superior temporal gyrus. And as we can see here, there's a little bit of difference. So we can see that the anterior cingulate cortex and the insula were more activated in the self-pain task, which is sort of expected because they do play a crucial role in the emotional aspects of pain and particularly self-experienced pain. And then we also found actually that the superior temporal gyrus was more activated in the empathic pain task. And this was also expected because it plays a role in empathic pain responses, particularly when you're observing pain through facial expressions. So at least we know that we're doing it right. And then
Yeah, so although there were some slight differences between like the self-pain and the empathic pain task, like as you could see in like this previous diagram, they were like very, very minor, but it does align with what we've seen in the literature so far. So regarding our second question, does loneliness or social connectedness at baseline predict activation in our regions of interest during the self-experienced pain versus the empathic pain task? So we looked at the ACC, we looked at the insular cortex, and then we also looked at the superior temporal gyrus. And across all of these, when we ran the MANOVA to see whether there was an interaction between the group that the participants were in, and then our loneliness, and then our social connectedness self-report data, the only relationship that we actually found was with respect to the ACC, and specifically in the self-pain task condition, not even in the empathic pain task. So the relationship here, the interaction here, it was significant in the ACC during the self-pain task, but take these findings with a grain of salt. We also ran a linear regression to see if we could replicate these findings, and it was not significant for the same sort of interaction that you see here. So what this tells us is there may be some sort of significance in this interaction and then the activation during the ACC, but it's probably a very weak relationship.
So as for functional connectivity findings, those are very disappointing as well. As you can see, they just look the same to us. Yeah. But this is like these are the functional connectivity matrices that are network block. And for more clear representation, I think if you guys can see the numbers are off by like 0.02, like between conditions. So and of course, like the edgewise paired t-test comparisons yield no significant result. And pass on to
Yeah. So then the second question for functional connectivity was whether or not it was associated with behavioral scores such as social connectedness or loneliness. So I just wanted to preface we did find significant results, but after like FCR correction, these results would not survive. So this is more kind of like a trend type of thing. So what we found was that functional connectivity with ROI pairs containing either cingulate cortex or the insula were negatively associated with social connectedness scores, but not with loneliness in both self and empathetic with empathetic pain tasks. So to put this like in a more like understandable terms, it appears that these connections seen on the screen between these ROI to ROI regions, apparently this decreased functional connectivity between these two regions is actually associated with increased with greater social connectedness scores, and the reverse is also true. So maybe it's like, oh, because these regions aren't as strongly connected, it stops like this type of like maladaptive like emotional regulation, leading to like increased social connectivity scores in these patients. But like there's a lot more interpretation to be done, especially with these like ROI to ROI periods.
So that was a lot, and where to go from here? We have a GitHub repository that we put everything into, as well as our data sets, our markdown files, as well as our analysis workflow and pipeline. And to bring all of this together, everyone's worked together on completing our training materials, including GitHub modules, Python modules, fMRI modules. And just to summarize our main findings here, we have the activation of the ACC and insula were higher in the self-experienced pain condition compared to the empathic pain condition. In the LKM group, that was that meditation group, higher loneliness was associated with lower ACC activation. However, this was not observed in the PMR group or that kind of active control group. The functional connectivity did not significantly differ between self-experienced pain and empathic pain conditions. And our functional connectivity and ROI pairs of interest, which were the cingulate cortex and insula, was associated with social connectedness in both the self and empathic runs, but these associations did not survive after correction. So, thank you so much, everyone, for your time. We'd be really happy to invite you.
Done a lot of work down.
Yeah, it was a big learning experience.
Yeah. So much work. I have questions, but
Be gentle with your questions. Please, no beginners.
So my name is Ruby. We have to announce our names, like our
Yes.
Yes. Okay. So what would you say like the biggest learning curve was for you when you were just
Can I say GitHub?
yes. Yeah.
Yeah, that too, but also just like where to even start, because we also had like a data set before, and then it was just not like feasible for like what we wanted to do, and then we had like panic, and we're like, oh, we have to find something else that will work and that has like enough avenues to like come up with like research questions. So
Yeah, figuring out what files are the ones we actually need to load as well. Like honestly, that was the toughest part, is just like where do we start? And then once we kind of had all of our like data like loaded into like VS Code, then it was a lot easier to do the analysis, but it was like literally those first few steps where it's like, what do we do?
Yeah, great job. Everything from beginning to end and trying to find a suitable data set with a really interesting task, the numbers as well that you use. Pipeline, so it was massive effort in a very short time, but a lot of work, great job, and a lot to learn to accomplish this. And also attempting to explain the results even though they're not significant, or why they're not significant, or what you can get out. So I actually have a question about the activation, the task activation. Could you address that?
Yeah. Which
The one with you identified the before and after activation, changes in activation.
the matrix?
Yeah.
This one. Yeah.
Yeah. So you identified regions of interest that you were looking at, seeing if they do follow. Did you look into anything that is opt out? What that was, why that could have come out.
Yeah, so some of the ones that are lit up, it was like very exciting to us, like, oh, like look, there's like a big difference here. But when we actually looked into the region, we were trying to see if there was like any literature to kind of support why we were seeing this. Like a lot of the regions that kind of showed up, and like a lot of them were kind of implicated, but there wasn't a lot of literature to really back up like why it would be lighting up for like one task or another. So
Yeah, I think it was mainly that like there were a lot of random regions that were popping up, and there's a lot of unknowns.
yeah, I filtered that out of the matrix because that would have like dumped. But yeah, like most of the regions that were popping up in accordance with our atlas were listed as like unknown. Like that was like the bulk of like the clusters we were seeing. So yeah, unfortunately not. Although it looks very like promising, like why is there such a big difference? Like we couldn't actually like figure out like why.
Yeah, especially with the tasks, the two types of tasks, they're very similar, and you have to think about exactly what is the difference and why it is.
it's very like interesting because also the literature for like neural correlates for like fMRI and activation, they just really state that empathetic pain and self-pain have really like similar overlapping like neural correlates. It's like great, but like that doesn't help us with like a certain like area or like ROI activation. So
So also, sorry, just also adding on to that, I think the one that pops out the most is actually like the fusiform gyres, which is for like face recognition, right? But it lights up in self-pink, but not like in happening. So that's like maybe they're just staring at the experimenter's face and want to like tell them that, oh, I'm in pain or something like that.
That's very interesting. Good job.
Great. Yeah.
No, but I guess yeah, we did that. Yeah, right. Yeah.
And I need you.
So for like the yeah. Yeah, so under this condition and then the other two, well, I think they were so good participants. Like, I mean, actually, that's what was so challenging. And actually, when we were trying to show down in this period of time, like with the same kind of like we couldn't even because like there was like maybe like one participant for their self-made. I love that. Yeah, so it was yeah. We were
Yeah, do we want to get started on the next one?
yeah, okay.
Okay, hi everyone, this is Nilay, and my project name is Functional Connectivity in ADHD, Group Differences in Predictive Modeling during Special Work. First of all, I'll start with background. I mentioned some of these in my project pitch, but working memory is a core component of executive function, and special working memory is often impaired in ADHD. And deficits in working memory contribute to inattention, impulsivity, and poor task performance. Also, difficulty sustaining attention in special tasks. So, neural mechanism that contribute to this is for especially special working memory tasks, typically dorsolateral prefrontal cortex for maintaining information, and parietal cortex for special processing. So, in ADHD, studies show that these areas show reduced activation. So my main questions were, I have two questions here, and one of them is, what is the difference in parietal functional connectivity between individuals with ADHD and controls during special working memory tasks? And the other one is, can ADHD be predicted using machine learning applied to functional connectivity data for special working memory tasks? So
Can you speak louder?
Okay, sorry.
From the back.
My main objectives here, my main goal is for this project was learn how to work with fMRI data. So this is my first time too, and more specifically, get more practice with the machine learning tools for fMRI data and learn how to compute correlation matrices and perform group comparisons. So the data set, actually in the project which I mentioned that I have 140 young adults in the data, but I think that maybe I cannot manage that lots of data, so I just want to pick half of them. And in my data for the analysis, I have 68 young adults, and 39 of them diagnosed with ADHD, and 29 of them are control.
So for the ADHD diagnosis, they use a scale, and if the participant meets the 6 out of the 9 criteria in either hyperactivity subscale or in attendance subscale, diagnosed with ADHD. So I got this data from Joel, thanks to him, and this is the file data for this. So the task was a computerized version of the special working memory task, and the data is a multimodal data. It includes both EEG and fMRI analysis, but I just focus on the fMRI. So this is the fMRI analysis workflow for me. So this one is the task-based analysis. So I start first with the selection of the atlas for the perspective brain. So I choose this atlas, AAL atlas, the automated anatomical labeling. So this atlas widely used brain perception scheme that divides the brain in 116 anatomical regions, and each region corresponds to a known anatomical brain structure. And the labels are really easy to understand. That's why I choose this atlas. To continue with the workflow. So first, using this atlas, I extract the time series by using Mathematics Masker. So then I also use, of course, the compound files for the motion control, and I compute connectivity matrices by using Pearson correlation. And I create a matrix for whole brain and for the frontal parietal regions. And then I conducted a comparison between ADHD and the control group by using t-test, and then I applied FTR function to them, and of course, then I visualize. So the results here, as you can see, this is the heat map of the results. So the red ones are higher correlation in ADHD than control, and the blue ones are higher connectivity, correlation connectivity for control groups than ADHD. So this is the how it looks, and this is how it looks in the brain, actually. And so after comparing this, and these are the significant results, but without FTR correction. Actually, I was too excited after that because I thought that I found some significant results, but then I asked one of the TAs, and they said I need to add some correction because with multiple analysis here. So this is how it looks in the brain. After using FTR correction, I have nothing. I think that's right. And yeah, this is the comparison part of the project. Then, even though I couldn't have find any significant results, I continue with the machine learning part for just the practice. So here I again upload the same functional connectivity matrices that I've found before, and I flatly reshape the matrix, and I used as a C classifier. Then I evaluate this with the k-fold cross validation, and I have now cross validated accuracy scores. So why I use this k-fold cross validation? First of all, I use random forest because I don't know why, but I use that because it's probably the basic one. And then I asked Erin about it, and she said since I use half of the data, it's maybe using k-fold is better. So k-fold is better with the limited data sets, and the logic behind it is like that, like they shuffle the data set randomly and split the data set into k groups, and for each unique group, so they this k-fold thing take the group as a whole,
and take the remaining group as training data set and a fit a model on the training set and evaluate it on the test set and retain the evaluation score and the model. So after that first validation, I have this. So it's like I of course predicted this result is like 50-50. And so I used 5. Okay, here. So also this is with the functional connectivity variables, but I use age and the gender again with the for the machine learning, but these are also like 50-50. So I use of course Git and GitHub, and I use Python libraries such as scikit-learn for machine learning and nylearn for fMRI connectivity analysis. For the visualization, I use Matplotlib and Seaborn, and of course I use Jupyter Notebook. So my deliverables here is are my GitHub repository. It's called scripts and data preparation, and I have, of course, as you can see there, figures and results summarizing performances of the models, and I have a really nice Jupyter notebook that includes analysis codes and visualizations. So, as a conclusion, what I learned here in this month, I started to understand the fMRI data, how to download, how to organize, how to manage the confounding variables. And also I learned functional connectivity analysis, how to compare two groups using atlases for brain translation. And this is my first time with the Python. So I learned also how to use Python debugging and practice with that. And I gained some confidence in working with neuroimaging libraries such as nylearn. And also I learned machine learning. I know that machine learning is used in feature selection, but I don't know how to do it. And also I don't know how to use this method for the classification, for the distinguish between the diagnose the, for example, ADHD and control groups. So I learned to explore the pipelines for diagnosis-based predictions. So for the feature directions, I want to learn the feature selection with machine learning, and then maybe using these features, I want to conduct functional connectivity comparison between ADHD and the control. And also I want to learn word processing, not related to that, but I think that maybe the harder part is that. So I want to learn this too, and also I want to upload this project to GitHub. These are my references, and thank you all for listening. So I want to thank TAs and Marian and Joel.
And great presentation. You mentioned that you used like half the data set to analyze for now. Do you think you would also observe the same type of findings if you looked at the entire data set? Or do you think increasing like the amount of sample size would help you with like the prediction model as well?
Yes, I think that probably I should do that. I think that it's not that hard to like, it doesn't matter if it's like 60 or like 100. But like I think that I'm not that, you know, I don't want to wait a lot that time. So probably maybe after FTR correction, I can see that significant results too.
Is another question.
My name is Kate. So I don't know, I kind of listed to choose say which ROI specific things from prior network did you choose? Or was it just
it was
based on the atlas.
Oh, okay, so anything that's in there.
Yes.
Oh. Do you think maybe if you focused on specific regions, that might be like maybe more related to our memory, you might find a different result, just because you probably have more power?
Rather than front of parental?
No, so like within the frontal striatum, if you just specifically chose certain brain regions that are more, yeah, I mean, that are more relevant for working on, do you think you would find stronger relationships? That would maybe serve.
Yeah.
Like it's a.
Yeah.
Do this or.
Thank you. Yeah.
Hi, my name's Ingrid. So for your K-fold cross-validation needs, I was wondering what other values of K you used and when did you see it?
Other cross validation?
Other values of K, like used 5, so did you try?
Rather than 5. I think they used 10.
Yeah.
5 or 10. I saw like that, but
Is anything 20, 100?
But your base sample is like 59, so you probably don't want to go more than 5 in the full. More than 5 is just in a full. Makes sense. What do you get? Sorry, I'm capturing.
I don't think there's any other questions, so
Okay.
Gia's group wants to get set up. Good job.
Oh, that's. Why, you got a full night's sleep?
Oh, right, actually, I think I read like that 1 a.m.
Yeah, that's the real gratitude is you can't function on 4 hours of sleep.
Wait, which one is your slides? I don't think there's one.
I'm just like.
Yeah, there's a video.
Yeah.
My phone.
Oh, okay, fine.
Charger? Was that all?
I have a charger.
Oh, no, it's
Oh, mine died.
in your group. Is Eleanor in your group?
Yeah. I think this might be.
Oh.
Yeah.
This is a project.
Yeah.
Oh, yeah.
Oh, what?
That is a good track.
Yeah, okay. Thanks, Julie.
After loading, is it?
It's, yeah, I'm buffering.
No, but honestly, if you.
Okay.
That's.
So hi everyone, our presentation is on increasing the accessibility of open access BrainH calculators. So BrainH is a brain age gap estimate. So just a brief background on what we discussed in our project kit. Is a model that estimates how a person's brain structure deviates from their actual biological age. And this is typically ascertained through MRI data or, through MRI data. And the reason this matters is because it's a tool that essentially allows us to better understand how the brain ages, which in the future could potentially be used to inform clinical outcomes.
So the main question that we wanted to address was how we can make these BrainHack calculators more accessible, and also more user-friendly for researchers that are using structural MRI data. And we wanted to do this by automating and standardizing the data preparation workflows by using R. So hopefully we can reduce the technical barriers to using these BrainHack calculators in research. So like I said, we wanted to increase accessibility by using these R-based tools, as well as creating public GitHub repositories so that we can promote reproducibility and encourage the use of these calculators.
And so for methods, we selected like an open access BrainAge, like BrainAge calculators. So it's like mainly from the Central Brain website. And then we selected 7 calculators. And we also choose to use the reproducible brain charts. Originally, we chose 3 data sets, but there's a lot of challenges just downloading them. So at the end, we chose HBN, which is the Healthy Brain Network data set to test on our scripts. And we wrote the scripts, and then so we'll talk more about each of the scripts later on, but mostly the scripts is to try to match the columns and try to make sure like it fits the template, so that it makes it easier for people to put in like data sets into the scripts, and then they can put into the template and then use the BrainAge calculator.
So just some of the tools that we used R to create the scripts for the templates. So we didn't use FreeSurf, but that is what we got our outputs from. And we luckily chose a data set that already was done for us. And then we also used Git and GitHub for version control. So in terms of our results, the first thing we did was create a GitHub repo to like house all of our scripts, and we collaborated on it together. And I think that was like a big learning curve for all of us. But it was really nice because we created a project and we assigned tasks to each other, and it was a really like streamlined way to collaborate. So kind of the first thing that we had to do for this project was download the data set. So like Eleanor mentioned, we used an open access data set through reproducible brain charts. And it's really nice because they have all of the their anatomical images already pre-processed. So they gave us, like they already had outputted FreeSurfer outputs. And more specifically, they also had data for each different atlas already on there, which was really amazing. And that's why we picked it, so that we wouldn't have to deal with the barriers of pre-processing any of the data. So the first thing we had to do was create a script to essentially create group data, because all of the look at my file structures, all of the HBN data was subject specific, and then it had 2 files, one with like the ASIC data that I'm sure you know about, and then the regional data, and all the regional data had every single atlas in one file, just insane, and a lot to have on one thing. And so we separated them, created a script that essentially pulls all the subject IDs and then also separates them based on atlas, so that whenever we do use the calculators, we can just pull the atlases that are specific to the calculators. So yeah. And then we're kind of gonna all talk about our own results, but on my end, I was able to output the brain charts template, but unfortunately brain charts did not agree with my template and is throwing errors at me. So that's going to be a next step, is trying to figure out why these errors would exist. And then I was also working on the Kaufman BrainAge, and I was also able to output a populated template for that, but they do have separate models that need to be downloaded, and so that's
So I was primarily focused on the Central Brain and Network BrainAge and network scripts. So the first steps were kind of just to clean up the code a little bit and make it more readable. So just kind of annotating that. And then one of the main challenges that I think Eleanor and I both ran into was matching the column names in the template to the column names in the data set. So since we were both having issues with that, we kind of decided to team up and work on the developmental calculator so that we could kind of figure out one and then apply that to the rest of the calculators. So we did manage to get that done. But towards the end of the script, it was throwing errors at us, which we still kind of had to troubleshoot.
So I mainly worked on the developmental and BrainHack calculators. So developmental calculator is around like 98% done with the script, but towards the end when we're trying to print out everything, it's throwing errors at us. And yeah, so mainly what we do is like first I did like a kind of like a directory, like make sure that everyone put everything in the like using the script, they are able to put everything in like the same folder and how they load all the data and everything and what we're using. And then we try to clean like the columns. So it's just trying to like match the columns. So I compared like the template columns to the data set columns and then trying to match everything and to kind of make sure that they are able to cross like match, but also the IDs as well as the demographic data because we need the age in the template. So it's just trying to match every Excel sheet and towards the end as I think for me, like for both of the calculators, it's like in the middle or towards the end, there's a lot of errors coming out. And so there's a lot of debugging and troubleshooting during the process. And hopefully in the next, I guess, 1 or 2 weeks, we're able to figure those errors out and really be able to output like something for the template and upload it to the calculators.
Yes, it's just a quick kind of summary of what we spoke about and what's been done and what we hope to accomplish at the end of this project.
So I just want to add that we do have a README file, so we kind of made that so that if anyone wants to replicate what we've done, they're able to follow it through the README file.
Yeah, and then in terms of skills learned, I know for me personally, this was very, very useful since it was the first time I've even heard of GitHub, as well as even like open terminal on the computer. So that was nice to know. And then also I used a little bit of R markdown in undergrad, but it wasn't very expensive. So it was very nice to kind of make or kind of design our own scripts and then kind of troubleshoot and see how to debug that.
So in the next steps, we are probably going to work on debugging the issues that we've encountered with the code for our calculators. And we're also going to increase our skill set with R, GitHub, and Markdown, and kind of, harness skills that we've learned throughout BrainHack School. So in conclusion, brain age models, they offer valuable insight, as I mentioned, like in the future, it could have very significant implications for both understanding neurotypical and neuroatypical developmental trajectories. But unfortunately, many calculators still remain underutilized. So because it's just a very jarring process as we've undergone. So our project was essentially not only there to ascertain data, but to help anyone who's trying to undergo this, like mammoth project. They're now able to, like go through it, hopefully with less headache and with less hurdles that we did. And this also addressed the barriers by developing like R-based wrangling scripts to kind of standardize things and make things a little bit more streamlined and accessible to everybody. Yep, and so that's the presentation. So thank you very much.
I see that you use the project function on GitHub, which is pretty fun. Have you tried creating issues and linking issues to projects?
Yeah, I actually made the issues first and then realized that I could.
Yeah.
But we didn't solve any of the issues. So now they're just sitting there.
You just create a blank one and solve it.
You're right.
Like first issue solved.
I think, yeah, I solved like one, so everything is okay. That's kind of cool.
They also have like this progress checking, like for the timeline kind of thing.
Yeah.
So it's like a learning process. Like there's a lot of things about Git I didn't know, and then now I do know, and there's a lot of things that we can learn in the future.
Like a great work on like because I know how much like moved you, especially how the work you did like the grade age computers and how like frustrating it is to get like it's actually accepted to it. So I was wondering like when like you guys are able to like finish because I'm sure you guys will figure out how to work out the errors eventually. I'm just wondering like how much time do you think it would save to like a struggling newbie who would have like no like
I think it would save a lot of time because like as Isabella and like Eleanor are having issues with column names, like to my master's also specifically like the central brain, it's really specific. And there's a lot of like if you run into an error with your column name, like it won't accept it. Like you won't get anything. So I think through this, if you kind of fix all those issues that we talked about, it would definitely be a lot easier for anyone else, you know, like picking this up, like they cannot have to go through those. They might obviously undergo natural like hurdles, but like the same hurdles that we went through, they might not experience that to the same degree, so that you know they can push the project, you know, like a little bit further.
I know you haven't like been able to extract much scores from it, and you were thinking about comparing them, but what would be your guess? How do you have a hypothesis or like a sense of how it might differ from each other?
Well, I think based on just training data size, I feel like brain charts should be good because it has the largest training data. I think it's like over 100,000. So I feel like with
And by good, you mean like closest to the actual?
yeah, closest to the actual age. And what we really want is for it to be very close to actual age so that it can pick up on any deviations whenever it has happened in individuals. So my hypothesis is that brain charts will work really well just because it has such a large training data size. And so I feel like intuitively that will really be better at predicting age in a wide sample. I know for my thesis, I want to look at a developmental population. For that reason, I think the developmental brain will work better because the sample size is because it was trained and tested on a developmental sample. So I just think it'll be more transferable. And then also, I think there's just a lot of nuances in the developmental sample when it comes to brain levels that it's kind of harder to be more accurate. So that's the only reason why I think that will perform better in comparison to the brain.
And I just wanted to add like so with my master's I'm using primarily brain chart. So there were some studies that indicated that brain chart has the like over brain age in terms of specificity and in terms of like really pinpointing the correct kind of brain trajectory. But I mean that's for my sample, which is a lot more advanced in their age, like they're about the age of 40, which is a lot more like precise. And like when the brain is developing, there's a lot more like factors at play. So that's just also something to consider as well.
If once you get the R scripts, right, just want to show that you've thought about like once you have the scripts, it's actually less hard than you think to do a Shiny app. And if you really want to like make it accessible without it would be like even easier to just tell someone to go to the website.
It will be also easy because you already have the R markdown. You can make it a portal markdown, and that it's basically the same thing, just changing RMD to QMD, and then you could have like a website with the Shiny apps on it, so people could just
no excuses anymore.
We'll be able to use it as a tool. And then you can just like, I don't know, add your names on the side and link it to Google Analytics so you can show how many people from where in the world are accessing your tools that you created.
But they couldn't edit.
You only have like an interface, so they can't change your code.
Yes. But they could also like sometimes with Shiny app they could download things from it. So just show it in the Excel sheet or something, but you can visualize it for them.
Yeah, I mean, I think we do want it. Maybe the only thing we're struggling with is like, we have to make sure that there's one that's specific to like how Christopher outputs things and the way they label it, then if there's different ways. But since we don't, that's kind of the stats.
What are your outputs?
Yeah, you guys are. Seems like that. There's no share.
You unmute yourself.
The slideshow.
We switched. So if you want to reshare, but share. What's happening? You get to reshare.
Hi everyone, thanks for joining us. My name is Ivy and I'm presenting with my teammate Chloe and Lucas. And our research topic is deep learning-based localization of EEG electrodes from 3D point cloud objects. Basically, it's an innovative solution, possibly a solution that can reduce the cost in mapping the EEG electrodes in current neurology practice. Wait.
Maybe.
No, it doesn't work. Wait, it goes out again. Sorry about the issue.
What did I say?
Slideshow.
You're gonna have to reshare this one.
Okay.
And then the
That's not showing there. This one, or maybe the Safari one, sure. Oh.
Weird.
Sure. Yeah, it's not changing. Or can we share the whole screen?
Yeah, that is the whole screen. I shared the whole screen in Safari.
Oh, doesn't work. Okay, I'll share the link to you.
It should be already in the Google Drive.
Mm.
We worked out of the Google Drive. You can stop sharing. Sure.
But how are we gonna control it?
Good question. How do we control? Just point every time. What's it if I blink like this?
Like an emeritus professor. Like, next slide.
Okay, next slide, please. Okay, sorry for the technical issue, and here we come to the background. As we know, when we do EEG examinations on patients, there's variation in the patient's head size and head shape, and also how we put the cap on the patient's head. Therefore, it creates differences and deviations from the results we would like to acquire. Conventionally, in modern practice, we use POHAMAS or videogrammetry to capture the coordinates or scan for the location of the electrodes. However, they're either expensive or they require manual labeling. Next slide, please.
I think you put up the wrong slide. Our pitch slides.
Oh.
Yeah, this is
There
not
should
the
be
right one.
images. Yeah. Sorry.
Yeah, we didn't, no one uploaded their actual.
No, there's two slides.
Oh.
There's the pitch slides and the.
I DM you on Discord as well.
There should be two. No, in the Google Drive. We worked out of the Google Drive.
Did you name it project pitch?
No, it's project wrap up. No, the last modification is May 17th, is that right?
What? But there, scroll down. Or all the way down or all the way up. What? We worked out of.
Can we upload it again?
Yes.
Where is it? Does it exist?
Did you upload it?
To that Google Drive.
Yeah, upload it again.
Wait, where is this?
But this is local, I think.
Yeah, it even says BrainHack School.
No, that's not the correct one.
Sorry.
Do you have it on your computer? Is that on your computer?
It's shared with the BrainHacks Google Drive as well.
Which one's a shared draw?
No. Is it in projects? Yeah, I DM to.
Oh, I put it in the general.
And the general as well.
I was like, looks kind of different.
That is the correct one.
We're gonna do it quickly now.
Okay, so the main question is it possible to develop a protocol that can automatically label the coordinates for the electrodes during EEG mapping and possibly use in future applications like MRI? Next slide, please. Our research question broke down into 3 parts. The red part is we haven't completed yet, but we automate electrodes localization and labeling, and we are halfway through of creating pipelines that can be possible replicating future usage. Next slide, please.
As described before, the data set comes from a multimodal data set that's available through the National Data Archive, but will eventually be on Open Neuro. The data type, the data set includes 144 participants, and each participant has a 3D scan head model wearing the EEG cap. We also have a subset of around 39 participants with the T1 weighted images with T2 weighted images while they were wearing the EEG cap in the scanner. And yeah, go. So do you want?
So the tools that we used, initially Joel manually selected and labeled the coordinates and created a TXT file. It will show the file later. And then we use the Open3D library to convert the surfaces and volume data into 3D point clouds. And there are two formats: the point cloud, which takes the vertices of the surfaces and the volumes and form the point cloud, but also the NPZ, which stores the labels as well, which we got from the TXT file. So, and then we took both of these, the labeled and unlabeled data, and we're able to transform them into the HDF5 format to train the deep learning model, and we use that the H5PY package. And then we are going to use the PointNet++ deep learning to train the model and test the data set afterwards. And also, because after the deep learning was trained, it will segment the electrodes. And to be able to get the coordinates of those electrodes, we are going to use a clustering algorithm, which is the k-means. And so we can find the centroids and then the coordinates of those centroids. And we also use the GitHub. We have the repository there, and where we inputted the initial scripts and Jupyter notebooks as well. Oh yeah, so this is basically our myth, like what we did this past few weeks. We started labeling the data and getting them to transform into the point cloud, and then HDF5 to train the point net. So we mainly focus on the data preparation. We started preparing the training as well, and also doing the script for finding the centroid after to be able to test the data as well.
And yeah, the parts that we're going to work on next are highlighted in red. So yeah, for initially, we did have the coordinates for all the mesh files. As you can see there, the coordinates correspond to specific electrodes and 3 XYZ positions. Unfortunately, we realized that all these were transformed into what's standardized for EEG space, so we had to figure out how to map it back onto our 3D models since they were transformed. So we worked on that for the first week, essentially creating ones with labels and then one we're going to test, data that we're going to test. Because we had to realign them and get them into the correct position, we ended up creating some tools for doing that as well that we added to the GitHub. And that involves like picking the fiducials and then realigning it onto the 3D mesh, and then the alignment app, which allows you to sort of translate and rotate the positions of those electrodes to actually map it out correctly onto the 3D mesh object.
So once we got the data and the unlabeled, once we have the files and also the TXT files, we were able to generate the point clouds, and you can see the lower image is just the point cloud. And then with the TXT file, when we have the coordinates, we created a radius around each coordinate to be able to account for certain difference and be certain that each coordinate is actually getting the electrodes. And so we created this radii to get all the point coordinates, or all the points that actually mean an electrode. So that's what we got shown in the second image there, and we were able also. So the red ones are all the electrodes in the scalps, but we also label differently the fiducials, which is the blue, green, and the yellow one.
Oh, and just to point out, the HDF5 files are particularly important because those actually store additional dimensions, including labels, as well as color information beyond the three initial points, and that's why we had to follow all these steps and get them and export them into that file format. For the middle one, it's just scalp versus electrode, but the last one is more showing just like from one direction to another. There's no real color, just more looks nice.
Yeah.
Oh no.
Oh no.
Oh, you know what? You're in your own Gmail, that's why. That's why.
Yeah, I share it.
Oh.
And
Oh. Oh, okay.
awesome.
Ooh, look at
This is a really cool video that we recorded. So basically, we mentioned the HDRF file, which is a hierarchy data format file, version 5. It usually stores coordinates and complicated data structure. For the H5 file, we need to cluster the centroids as we previously used in point maps. It's all the dots. And we use k-means clustering here as a method. Basically, it means the distance between clusters of points will be smallest when it's referring to one centroid. That's the centroid of a cluster. Here we can see blue is a cluster, yellow is a cluster, and the green the cluster is the centroid is in the middle. Therefore, we generate this rotating video, and each color stands for a protrusion, which is like basically electrodes area, and the red point is the centroid, basically it's the electrode. However, there's some false positive here. As near the neck, we can see it labels like 2 or 3 electrodes. We need to use the manually labeled coordinates to use a template to remove it. However, we haven't done this step yet. Next slide, please.
So yeah, in terms of deliverables, we've created a GitHub repository, including our scripts, and we're hoping to continue organizing it and have something that you can call as a package, the deep electrode mapper, as well as some deep learning notebook example tutorials on how to do each step. We also include the PointNet2 package. A lot of when I was setting up and installing, we realized that PointNet2 is like 7 years old now, so a lot of the Python tools as well as TensorFlow has been updated, and we have to actually alter their scripts to work with the newer TensorFlow. And in terms of challenges, we ran into a lot of challenges while doing this. We learned a lot about how to work with 3D files, as well as what it takes to actually train a deep learning model. One of the issues that I ran into is our annotation is sort of like a quick and dirty annotation by mapping the electrodes and taking the radius. Ideally, we'd have a tool that would be able to sort of annotate which ones are electrodes, and then specify like, oh, this is hair, this is actual scalp, as a way to better train the deep learning model to segment the 3D objects. Another thing that I realized too is that a lot of the pre-trained models on the internet all focus on objects. There's nothing that's very specific to like segmenting bodies or faces in 3D specifically. And so we couldn't take a previously created model and fine-tune our data on the pre-trained model. So we're having to actually do a training data set from scratch. So yeah, you have a lot. And next steps, we want to actually run the training on PointNet. and hopefully create a model that we can upload online for other people to try to use and test. We're considering creating synthetic data to increase the training data set. Since we only have 143 participants, we could potentially improve a model. We don't know if we need to improve it yet, it's never run it, but that's something we're considering, since a lot of the 3D object model, like what we've seen online, requires like thousands of data sets to train. And then we're going to test it on their MRI data and potentially create a 3D model annotation tool that's easily useful in Python, since a lot of the ones required that you upload your data to a server, like someone else's server, or if they were not open source, you need to pay for that annotation tool. And here are references, and if you have any questions.
Sorry, one question. What is the input of this point net? Is it just for what the exact input?
So the endpoint is, if you go back to like slide 12 or something.
Slide 12.
Or
Okay.
So the input is this data file that contains the coordinates. So basically, we simplified our 3D model to be specific points. And then, so that file contains the 3 coordinates of each point, as well as a label that says whether it's scalp or electrode. And so that's what we're training the model on, is to be able to differentiate what is scalp and what is electrode for each point.
Okay, so you have XYZ for the electrode locations, and then 2 class scalp and electrode.
Yeah, and then we're hoping to also eventually add our RGB color information, because there are so we can contrast like hair versus electrodes as a like a 5th dimension.
Okay, and I see that there was like segments around each electrode as an output, like different segments with different size. How those was generated? Is it the
clusters?
the clip that you shows?
Oh yeah. That was on the whole, we did cluster on the whole data set to show as an example, but this is showing like there are clusters that don't represent electrodes, and part of the deep learning is we're hoping that by labeling scalp and electrode, we're able to remove the portion that is labeled as scalp and then cluster, like have more precise clustering occurring.
And how this segmentation was done?
Basically, we used k-means, and since there's 126 electrodes, we set the k to 126. This means we extract 126 area and find the centroids of it. And the reason why it shows the back area that has the centroid, is because we trained the whole data set instead of training the data set with the coordinates that we manually labeled. That's one of the fallback of our research. And that's why I previously mentioned we need to use a template that has the originally labeled coordinates and remove it from this video.
And what is the input of this k-mean clustering?
These are from the point cloud, and each point basically represents the area that the electrodes taken. And it's like a 3D mapping of something. Basically, it's like scanning of someone's scalp, and then each point represents the area that a scan represents.
So we essentially skipped the step of training the model and just ran k-means directly on our data set.
Which is kind of cool that it works.
That it
works on the top of the pen.
Yeah.
Yeah.
So we essentially skipped the deep learning just so that we can create the scripts for the centroids. But yeah, as you can see, it does a pretty good job for some of it, but not all the electrodes are on there.
I make me wonder too, like classic free server, old school free server. There's a lot with just the curvature, like whether like it defines the shatters versus solids based on like the curvature of the mesh. I mean, like concave versus concave. Yeah. It makes me wonder if we could actually also just
also label that type of a.
I forget how it gets that curve stat. I don't know how it's calculating that curve stat, actually. It's magic, it exists. And then
It has greens and reds all over the freezer box.
but
Oops, so many questions.
oh yeah.
My name is Kayla. Great presentation, really interesting. I'm more curious about maybe what was the inspiration behind working on a deep learning model or like even just with EEG, just because there's so much data to choose from. So why this particular project? Like maybe past experience with EEG or maybe no experience with EEG or
Basically these data are from Joy's lab, and the reason that we decided to use a deep learning model because everyone's head shape and size is different, and how the technician put on the cap is different. So we want to minimize the cost and labor to mapping those coordinates automatically. Therefore, we're thinking about, okay, maybe a deep learning model can help current neurology practice to mapping the coordinates better.
And for context, there are other tools for labeling, but it can range from 30 minutes to an hour to label a data set. So in a sense, for example, the behemoths you can label in person for participant, but it takes 30 minutes to do, and you're taking participants time to do it. When you 3D scan, it just takes a minute to scan it. But then
There's still already so much time spent on like fitting it properly, and
Yeah.
every electrode is like lighting up properly, so you don't wanna.
yeah, you want to reduce the overall time with participants. So this doing a 3D scan does do that, but then when on the person who's processing it, it takes 30 minutes to an hour to label each one of the electrodes. There are tools that do semi-automatic labeling, but then you still have to like specify certain labels and then hope that the semi-automatic will label it correctly. Otherwise, you have to go back and edit each one. So we were hoping that maybe using a deep learning model can label all the electrodes like better, like so it's fully automatic. So you just have to like load it in Python, run the model, and then get the output. But it's way more complicated than that. So we learned, and I personally also wanted to learn about deep learning, which made me realize that it takes a lot of data to train a model, thousands of data. So.
Amanda?
Yeah, I have a question about like training the deep learning model. I'm wondering, is this something for this project specifically? Is this something global or do you require like access to stuff?
We haven't gone that far yet. I was able to install it on my mobile computer, but I also have like a Nvidia graphics card because I didn't want to do modeling for lab. Although we don't know how long the training is going to be, but we have a pretty small data set, and depending on like the booking size, which I still don't know what that means, like if you use smaller books, then the training is a lot shorter. But then again, we saw a publication that did this in MRI and said the training data set took 2 weeks to do on muscle. So you can
Just curious, can you go back to the video? I'm assuming that I'm assuming like video is showing like a scalp, right? But it just occurred to me that it seems that there's electrolyte covering the face as well.
Yeah.
Oh yeah.
So that's part of why like we want to we think it'll work better once we're able to separate scalp and electrode and just remove all the points that are labeled as scalp. And that's what we're hoping that the deep learning tool, once we put a bunch of points into the tool, it'll come out with the labels, and then we just remove scalp and then run k-means on only points that were labeled as electrodes. And hopefully that will this already looks great. It would just need to actually improve it and remove the unwanted labels.
Right.
Yeah.
Okay, we're just gonna get started then. This is our BrainHack project, which looked at exploring ADHD connectivity through feature calculation. There's a member of our group that isn't here today, and Sam's gonna have to leave soon, but he's gonna just run out, but we'll just get to it. Yeah.
Yeah, okay.
You wanna just get us started? Oh, sorry. Go ahead.
Perfect. And for the background. So ADHD is characterized by attentional difficulties, impulsivity, and hyperactivity. And so previous research has suggested an inter-network functional connectivity between somato-motor network and dorsal attentional network, which is uniquely impaired in individuals with ADHD. And so there's also differences in the hyper or hypo connectivity, so reduced connectivity or a lot of connectivity, network modularity, whole brain functional network segregation, and various subtypes of ADHD. Yeah. And so our main question here, how does region and network connectivity differ in adults with and without ADHD? Okay,
That's my job. Okay, and so our objectives for this project were to 1, practice data wrangling with large data sets and visualizations in R. Learn to use Python for data analysis and visualization. Use GitHub for collaboration because none of us have ever used GitHub before. Become familiar with a new data modular or modality. Some of us have never seen fMRI data before, so it was fun to explore and work with. And then finally, we initially set out to attempt core periphery modeling in a widely used data set. I don't think we really accomplished that, but that was one of our objectives.
It was too hard to do.
Yeah.
Very ambitious.
For the scope of
we did it. We did it. So the MRI data set, it's the NKI Rockland data sample. And so all of the MRI data was pre-processed using the virtual brain UK biobank pipeline, the Schaefer-Tin combination atlas for cortical and subcortical parcellations. The resting state fMRI data were pre-processed with fMRI subpipeline, which uses the FSL's toolbox, and then that includes the motion correction via realignment using exploit. And so for our project specifically, we had 54 participants. So we had 12 males and 42 females, age ranging between 19 and 65. In our ADHD group, we had 18 people, and then our control had 36. So we had 2 controls per ADHD. We also matched for presence of traumatic brain injury. So no one had traumatic brain injury. And then we also matched for sex and gender.
Yeah, so in terms of our approach, we first, because the data was already pre-processed, we started by filtering the relevant demographic data that we used to create our sample. So like we match for age and sex. So we chose all of the subjects that had an ADHD self-referred diagnosis, and that's why we end up with 18. So then we just matched the control, 2 controls per case. Then we created the average weighted adjacency binary adjacency matrices. And after that, we calculated the centrality measure. So for these, we chose node strength based on the core periphery model that we were trying to attempt.
So we did like a half.
Yeah. Then also chose the participation coefficient and with the module C-score. So after that, we tested the difference between in the features between the control group and the ADHD group, and categorized the regions of features based on the 7 networks for visualization. And then at the end, we did feature selection using random forest. So we'll talk about the tools. The tools that we use, like we said before, are in our studio, Tidyverse Group, and Digital 2.
Actually, among other ones too.
Yeah. Yeah, and then for Python, we use it through a Jupyter Notebook in Visual Studio Code. We use the Brain Connectivity Toolbox and MindLearn, and yeah, we put it all in a GitHub repository.
So this is just a really brief snippet of what some of the results look like from filtering the demographic data.
So on the left here, we have like some of the code that we wrote to get the CSV file. That was what you can see there on the right. So we have like different types of ID codes because the demographic and the brain data were like split differently. Shout out all the TAs that helped us with that.
Yeah, fun fact, our data set was in a cube form, so this is the first time that we ever worked with a cube, and yeah, this is a big part of 2 and 3 work, I think.
And then again, just also including in ADHD diagnosis or not. And then jumping in more broadly into like the overall functional connectivity matrices. These are the functional connectivity matrices of the ADHD group as mean and the control group. And we don't really see, or at least me, mine, like the very small neuroimaging background, like I don't see major differences. There obviously are a few. I see like a little bit of a difference in the ADHD group from the control group, but not anything super great.
Sorry, I have one question. Yeah, what is this bomb? There's the little bombs here.
Yeah, I think that it's good that you're on left, right.
What is that?
Because it's good because I'm pretty sure that the ROI list goes left, all left hemisphere or ROI in the contralateral, right? And if your ROI, they create a diagonal when you actually get the contralateral regions.
So I think it's just repeating the same part, right?
No, because like the brighter like the diagonals with itself,
yes,
or with itself or with thing, then
this one's interhemispheric.
Yeah, that's your interhemispheric connectivity.
Yeah, I understand that it's diagonal, but there's a bump.
I talk to you here.
I think it's just like the
activation.
I'm just curious.
Yeah, I think it gets because the where's the subcortical? Yeah, I think some of the networks at the very middle are a bit weird.
I think they were half, kind of like 3/4 of the way through.
Yeah.
Of the
Yeah, I think
the bumps might be the laterality because if you look at it, it's between the left and the right hemisphere from like maybe 100 to 200 RIs. If you look at the one within the hemisphere, they're pretty straight. So there could be some laterality.
In general, it's very pretty network structure.
Yeah. Yes, like what you wanted to.
Yes.
You want to see the blocks.
Yeah.
Well, thank you for that. Okay, and then these are just some brief results of our centrality measures. This was node strength in each of the groups as well. So the control group at the top and the ADHD group at the bottom. And again, this is just based on the mean strength. Those are those colors on the brain. But then we ran some t-tests to actually pull out significance of ROIs, and that's what you see here.
So one was significant, but after the correction,
400. Yes. And then we have another one like this as well. So this is the participation coefficient. Sorry, Ruthie. We did try. The participation these are the participation coefficients. So again, the same thing with the control group up top and the ADHD group at the bottom. And we did the same thing. We ran independent sample t-tests again to pull out significance. It's the same thing as before. So we had some significance and then we adjusted p-values changed.
Yeah, I think there might be something off.
Participants,
which we will get to.
Yeah, so yeah, I can comment on that. So we did check the so for these, everything looks mostly yellow. So that's because most of the values were positive. There were only like 4 for controls and 2 for ADHD that were negative. So that's why the colors are skewed.
Yeah.
So everything was like positive, and then just these ones were so.
What does it mean to make this?
Oh,
no,
sorry.
I think it's there's some error in that specific version. That's what
Yeah,
we
and
have
the ones
this-
that went negative.
Yeah, and now it's just like the whole color scale is driven by that specific version, so they couldn't see any.
We did adjust for the two, you know, there was like the 90 something. So we changed it to like -3. Like instead of being -90, it was like -3. But there was like -3, and then -2, -1, and that was it. So there was no, everything else was like 0.9 point. So it was like on the opposite side. So, but yeah, there might be something.
Yeah. But potentially you will want to run it instead of just changing it to -3.
Yeah, well, just for the plot, we were trying to see how would it change, and it didn't really change much. So yes,
Yeah, I could do working on it. See what's up right there.
And then here we are again, more of the same images. But this is the within module Z scores. And with the groups there and the mean strength of those. And then we had a lot of p-values first come in as significant for various regions of interest. And I didn't mention this before, but we tried to color code to the best of our abilities these regions of interest to the networks that they are a part of. But again, the values are not amazing. But you know what? Sometimes that's so meaningful work.
Yeah, and then, so like she mentioned, we got the ROIs and did the groups by set of networks. So we have the visual, somatomotor, visual attention, limbic, control, dose attention, and then default. So it was my first time making like these kind of plots. So I made these lovely timeline plots.
Yeah, so we only did it for node strength.
Yeah, we only did it for node strength and not for the purchasing coefficient and
because of
time.
time.
Because of time. But in the future, we might.
Yeah. Okay. Yeah, so in parallel to that, we also run random forest to kind of highlight which regions of interest were more important in defining the two groups. And so these are the results for the node strength measure. And we also tried to color code them based on the networks. We didn't see it. Yeah. We didn't see like a pattern per se, but yes, it's something that we can work on. So this is for node strength, and we have another one for within module C score as well, and then another one for the participation coefficient. And at the end, we did also run them all together. So these are the results of like the three centrality measures all together in one course, and kind of like it seems like the within module C score is the one that having
Contributing
yeah.
important.
Yeah. That's good. Oh yeah, and then sorry. So the overall accuracy of the whole model was like 0.71. For the other one, I think was similar as well, like around 0.7 to 0.6.
Thank you.
Yeah.
Yeah. 71. 86 and 71. Oh, sorry. So for the deliverables of this project, like many others in the room, we ended up creating a GitHub repository with a bunch of different files and resources that we used to put the project together and tried to create some pretty figures for you to look at. So the main conclusions, BrainHack. What did we achieve? Grace, what did we achieve?
Well, we learned some new skills in coding in R and Python. And we also used GitHub effectively as like in a collaborative nature, which we've never done before, which was really fun.
Yeah, and it was interesting also because Loretta mentioned she'd used GitHub before, but only alone, not as a group. So it was nice some of us were introduced to it for the first time or introduced to it working in as a group. We did encounter many challenges from accessing instructions for how to run our interested like models at first and not being able to, as well as running into accessing specific parts of the data that we want to use if we wanted to like continue this project in the future, which we do. We're just kind of waiting with some hurdles that come with timing and research, as well as small coding challenges in both the brain and behavioral scripts. So just running into things that I'm sure everybody coding runs into every day. But we do plan on refining this project and continuing with it outside of BrainHack by exploring other centrality measures as well, accounting for movement, like I mentioned as well,
And possibly in the future, writing a manuscript if there's anything worth writing about. But yeah, just refining our skills as researchers.
Nice visualization.
Yeah.
I appreciate
Yeah, I
it.
was gonna ask.
Did you say? What did you say?
Did you say?
What did you say?
Go ahead.
I was gonna ask, Joel here, I was gonna ask what art you use for the visualization?
Gigi said.
Yes,
Yeah.
I don't know if you guys are holding out, let's see.
Yeah, there's another one. This presentation, I really liked it. I was wondering if you did any study taking for your analysis in terms of like either ADHD diagnosis or even stuff like sex, considering the female population higher.
Yeah, I mean, I think that's like pretty popular in research. There's often like a major sex difference in the size of participant samples. But we just like used what we had, and then the subtyping for ADHD, we didn't use it, but I'm not sure actually if it's in the data. It might be. So we, might be able to explore that in the future as well.
Interesting.
With the strange pattern in your participation coefficient, it might be, you might want to go back to first check your connectivity matrix and then to check the connectivity matrix after you go there, because you take the top 4%, right? You might want to check what that matrix looks like. It could be that it looks too empty because you're also removing the negative values. If you remove the negative values.
No, but wouldn't that affect all the value measures, not just that one? Because we use the same group.
It could be. So it could be like all over. And so can you remove the negative values, the negative edges? I don't think so. You might have to do that. So you take the top 4% and then you remove all the negative values, replace that with zeros.
Okay.
And also before that your R values need to be Z transformed. And then you put that into the brain connector. Okay. Option. So
Yeah, we did
this must
replace
be
like
where we
the
were.
the zero diagonal. We did that part, but
yeah,
other than that.
Yeah, so those might be the 2 steps that's giving you like weird results, and maybe that region specifically like is too sparse or something. That's why the PC is really strange, like an outlier.
Yeah.
But I think it's interesting that you see, if you go back to your random forest results, the second, this one.
Oh, yeah.
So you see the default and the visual are coming. You do see some patterns here, like visual defaults are coming out on top of product too. And in the previous one, I think you have some limbic.
Yeah.
This one,
Makes sense for the ADHD.
Interesting. Yeah, and you might be able to see more, like to see the pattern in a better way by just finding those spots in the brain to visualize them.
We're also thinking of like using those regions that came out as significant before the adjustment to just for the random forest, just to see. So we didn't get to that yet.
Feature. So didn't you try that and you found a worse accuracy?
No, so when I was doing the feature selection only with that random forest, sometimes it went from like 65 to like from 71 to like 69 something, like after the feature selection. But that was just with the random forest, not with the like selected features. So yeah, we can also like play with like the different metrics. Like I didn't play much with like the different metrics of the random forest modifier, so
Also, like a clarification question, is this just for the ADHD participants or is this the entire?
Yeah, so the random forest is giving the variables that, the features that are most important in classifying your groups. So because we only have two groups, yeah, that will be like the ADHD group, like the.
One more question. Is this the next thing say data?
Yeah, this is our state data.
So, I'm not sure. Did you use a specific atlas at first or the?
Yeah, we did and put it here. Oh, yeah, we did. We with the 420 parcellations. Thank you.
No, I thought it was 5. No, that's
Okay.
Hello, good afternoon, everyone. I'm Ingrid.
I'm Amanda.
Today, we'll be presenting our BrainHack project, Age-Dependent EEG Patterns for Predicting Treatment Response in ADHD. So, some background. ADHD is one of the most common neurodevelopmental and psychiatric disorders. ADHD is characterized by significant neurophysiological differences that can be observed in EEG recordings. Electroencephalography, or EEG, is a powerful neuroimaging technique used to extract features and patterns such as I am.
I'm not joining the group, the Zoom.
This one is unmuted. Unmuted. You won't.
Oh, because.
Okay. Okay. The neuroimaging technique used to extract features and patterns. From EEG, we can extract the power spectral bands, where there are 5 bands that represent different functional characteristics. For example, theta band is associated with deep sleep, and the beta band for awakened concentration. And patients with ADHD typically generate an abundance of low frequency delta or beta brainwaves, and have a shortage of high frequency beta brainwaves. A promising development in EEG research is the use of AI and machine learning as an advanced signal processing tool. There's also age-related changes in EEG in ADHD. However, there's a lack of personalized treatment recommendations based on EEG patterns and age for individuals with ADHD. So for our project, we wanted to answer, how can we use EEG patterns to predict treatment responses for individuals with ADHD across different age groups?
So for our objectives, our main objective is to learn how to process EEG data in different machine learning methods. And then our second objective for this specific project is to assess age specificity of EEG-based treatment prediction by developing an age-dependent model to predict treatment response across different age groups. And then we want to identify EEG signatures of neurofeedback treatment response across the developmental stages to determine if the responders will show different EEG signatures at different developmental stages when compared to non-responders. So for the data that we used, we chose an open access data set called TD Brain Database. It is an extensive clinical EEG data set with more than 1,000 individuals collected over 2 decades with a wide range of age range. It has broad resting EEG recordings, different personality data, demographic information, and behavioral measures.
We chose it because not only because it's publicly available, but it also came with a Python pipeline for artifact identification. And it has ADHD cohort and a sub-cohort in the ADHD samples with treatment response for neurofeedback treatment. For our method, we started with the raw data that we download from their website. It contains demographics information and some metadata include different behavioral measures. This cohort has 204 ADHD samples and only 70 of them had treatment data. We also downloaded EEG data for all the individuals. And then for pre-processing, we first ran their pipeline to identify artifact, and we also noticed that their pipeline only identifies the artifact but not removal. So we had to write our own code for the artifact removal, and we did epoching and feature extraction for those band powers and different power ratios. And then because we had a lot of features, so we had to select different features, select top performing features before running our random forest model. We used Bootstrap to first determine the optimal number of features to be put into the model, and then we selected the top features being selected by Bootstrap. And then to build our final model, we also used Bootstrap for validation because we had a small data set. It was only like 17 individuals, so we thought cross-validation is probably not the best option. The Bootstrap gave us a recommendation of 60 features to be included in the model. So we selected top 60 features for our final model, and we used 7 Bootstrap iterations for model evaluation. There are some missing data in the feature being extracted, so we did median imputation per iteration for the evaluation, and the final model had a value trees and used top 60 features. And then after selecting the features, we tried to do some interpretation and ran some age-related analyses to see if there is any different patterns shown for different age groups across responders and non-responders.
We used various tools for our project for like numerical and signal processing. We would use NumPy and SciPy. Signal processing is free. So we use a combination of Python and R. R we Parrot and Random Forest, we do Plot. For collaboration, we did Jupyter notebooks and then started with the laptops and have the GitHub repository. So we created an organized comprehensive data set merging the EEG demographics and the features for participants with ADHD for our machine learning analysis. And features were extracted from each electrode and for each power band. So for example, we have P1 electrode, delta, power.
Yeah, this is to show the distribution of our sample. As you can see, we have a quite imbalanced data set with a lot of responders, but only a little bit non-responders. For example, if you look at the oldest group, it only has 1 responder. So it makes the analysis a bit challenging, especially when running the bootstrapping, because some of the samples could have no responders in the sample. And on the right is a graph showing the missing data pattern. We had around like 1% of the missing data for top 60 selected features. Most of them, I think, is a result because of artifact removal. So we did median imputation during like a random forest and bootstrapping process.
So we built our random forest using the bootstrapping validation with optimal features. So on the left is the model's accuracy performance with an AUC of 0.865, indicating strong predictive capability. Our RSC curve shows discrimination between the responders and non-responders. And then on the right is the feature important analysis listing the top 15 features. So for example, at the top, most important predictor was the delta wave measured from the central region. And interestingly, we did find that age was one of the top important features. And so this importance could suggest that treatment response prediction relies on a combination of spectral power and between different brain regions and again.
So we found that the frontal lobe electrodes were presented in top features identified by the random forest classifier on the right. And then that there were low frequency delta or theta brainwaves represented in top features. So for example, going highlighted here from the FP1 electrode delta relative power. And from literature, we know that patients with ADHD typically generate an abundance of low frequency delta or theta, and the EEG may show that person with ADHD has excess slow delta or theta in the frontal lobes of the brain. So we were able to find from our random forest classifier that it did discriminate as the delta relative power as an important feature between responders and non-responders.
And then we did some age-related analyses trying to correlate the identified like the most important features with age to see if there is any correlations. And we also did a age variability analysis to see if any features varies a lot between different age groups. Those being those in the green box are the overlap between highly correlated age features, and those are highly variable between different groups. And then we also plotted out the patterns. Patterns with those overlapped features from non-responders and responders. A note to the slide is that I don't think the non-responder pattern is very reliable because of the extremely low sample size, especially for the oldest group. We only had one individual, so it's the data from that one individual. From here, there isn't really a very significant pattern or difference that can be identified from non-responders and responders. On the right is just a different representation of those features in line plots. So if it's possible to include more sample for each age group, we probably could identify a significant difference, but for now, this data doesn't really show a huge pattern for different age groups.
By the end of this project, we were able to develop our workflow using Python and R, and also the TD brain code that was provided. And by the end of this, we'll have individual reports. In conclusion, our random forest model achieved AUC of 0.86, which is a potential overfit, but this can be helped with validating the model on a separate or larger data set, which would be the next steps. We also concluded that age is emerged as an important predictor with several EEG features showing strong correlation with age. And there are also age-related patterns that provide a multidimensional neurosignature for predicting treatment response for neurofeedback treatment. So the limitations we faced in this project was the small sample size, the data imbalance, the missing data, and that only neurofeedback treatment was data was available to examine any other ADHD medication.
As a recent art project.
Great presentation, thank you. I didn't really understand how to define the features, like is it average number of different bands over the entire equal? What was the length of the equal? I didn't like I received, maybe you explain.
So it was 2 minutes of eyes closed and eyes open from the data, and then we epoch into 30 seconds, and then
it just what
30 seconds.
30 seconds, okay.
And then did power spectral instruments, and then we got the total power, and then we would do relative power. That's what we mostly relied on. And then from there we would make ratios and combinations of them, and we had our data set. We did that for each of the channels. We ended up having like 600 features with the different combinations, and also using these as well.
I was just wondering what the treatment actually was like for this patient population and how they differentiated between like responders and non-responders.
The treatment itself is more about neurofeedback treatment. And to differentiate between responder and non-responder, they had a pre-treatment score for hyperactivity and attention deficiency. And then they also had a post-treatment score. So if there is significant improvement, I think it's defined by improvement by 30% in the original data set, it would define as responder. And if below that is going to be defined as non-responders.
So when this neurofeedback treatments are not
actually so what is the neurofeedback treatment?
From our reading, they would like stimulate. They would do the resting state recordings first, and then afterwards they would stimulate their brains, and then we do another recording after and see what the changes were.
Usually involves like real-time feedback of like being able to see like your alpha wave or something, and then asking them to like dampen it by doing something, and then so like training them to modulate their own. It's crazy.
So that's interesting because if you go back to the features that were identified as important, there was one as alpha center frequency, correct? Here, yes, CPZ alpha center frequency. So if they're depending on, I'm not sure how they did the neurofeedback, but if they're averaging an alpha or like choosing a band or how they're showing the alpha band that they're modulating, and it's a feature according to the two responders versus non-responders. So then there might be the center frequency, there might be an actual difference in the alpha center frequency between those two groups, and that they're not targeting the center frequency of those the non-responders, which might have this. There's a lot of clinical applications where the alpha, so the alpha frequency is different for those who go over. So it could be because they're not actually targeting the personalized center frequency. If so, have you looked at if there's actually a difference between those two groups in the top features?
Yeah, I think in the original data set there is different protocols being defined, but we didn't specifically look at it. And we also include the protocols into the random forest model, but I don't think it's being selected as the top features, so it's not like presented here.
I'm sorry, how do you normalize the features before you move it to the model?
Yes.
This the power is different, the scale of power in different electrons.
You mean the absolute power?
Yeah, the power of the electrodes are different. Also, it could affect the numbers.
Oh, you mean normalization like for each electrode or?
Yeah, I mean, different electrodes have different range of power. So that would be affect the nodal decision if like on some regions have more power relative to others.
So it tries to rely more on relative power to
in the future, if you continue looking at this, I would also recommend source localizing and looking at source CG.
I'm sorry, could you speak louder?
Was there like a region of different age?
If you look horizontally, the this one is the most significantly different ones is the delta F4 delta absolute value.
F4 so frontal. But we do know from EEG it's very age dependent. So like you will like as you get older decrease brain activity. So we are seeing like a downward trend in the C3 at the top of the left. The R plot, it's like down in age, and some of the bottom
bottom right corner is right there. Recently. Or even honestly like whole brain, as you age, like it's gonna be detected by the EEG. We usually want to go just investigate the entire brain, but just from literature, we're going to pay attention to the frontal lobe. Let me show you everything today.
So just for the age, I think because like the delta band is at like the furthest, I mean like the end that's like closer to zero in the power density spectrum. So like the change in absolute delta power is actually like it could be like largely due to the change in aperiodic signal. So like what we know about the aperiodic signal is that as you age, the aperiodic exponent, so like the slope of your power density spectrum actually gets flatter. So like we know that as you age, you get like the flatter slope. So that might be why the delta absolute power is like changing with age.
I mean, I was just I saw I don't know. We're just looking at differences, and one of the ones that we like the prior, and so also just it also just has to do with about
I think there's like a very recent systematic review about like how which shows that throughout like the early development stages, like the absolute delta power just decrease or increase. I mean like there's just a difference.
Yeah, it was fun.
Yeah, I feel like maybe talk about this in your project pitch. For the age groups, how did you determine that age range? Was that like already set by the people who published the data?
No, we just try to have the same number of individuals in each of the group, and we just divide the age range by this number of people in each group, and then this is like the distribution.
Can I make a suggestion just because there are so few non-responders in the older age group? It might be obviously it like makes sense if you're trying to, I don't know how large the age range would have to be to really pick up on differences. But it might be helpful to look at the mean age as opposed to like kind of like arbitrarily like categorizing it into like these different like age ranges, and then maybe look at like a standard or multiple standard deviations above and below the mean to kind of get a better sense of whether there are differences in the responder versus non-responder category, because it's tough like this when you have such a skewed distribution, you can't really like pick up on those differences. So maybe like a suggestion for the future, you might be able to pick up on some differences between the responder and non-responder group if you like look at the mean instead of like categorizing it into different age ranges. Okay.
Yeah.
Cool. So I'm Fardeen and I work with this with Sam. Unfortunately, she can't make it here today. But what we looked into is EEG-based monitoring of cognitive states in athletes. So a quick background, EEGs, and then we chose athletes because they operate under high pressure conditions. So we thought it'd be cool to see if there's any way we could help them train based on like their neurals instead of just a physical kind of standpoint. So we looked into these 4 bands, alpha, beta, theta, gamma, with these ranges. And our objective was to analyze EEG frequency band dynamics in athletes performing motor and mental tasks to detect whether we could see any changes and if that could help with their training and their performance in the game. So in terms of data tools and methods, our collection was we extracted the raw brain waves using a Muse device during live motion recording. So Sam basically collected it from golfers, and this was during their swings. And we recorded the subjective performance on a 10-point Likert scale during these trials. So this is whether bad shot 0, good shot 10, and they just recorded each shot on that range after each shot. So in terms of data processing, we had a sampling frequency at 256 hertz, and we did change the filter from 1 to 40 to now 1 to 50 to account for larger gamma band frequency. And we had to go through processing, so artifact and blind data removal. In terms of analysis, we mostly used M&E from Python. So we did segmentation. So the EEG signals, we segmented it based on what type of task they were doing. We did frequency analyses for each of the 4 bands. We did topo maps, and we ran a correlational analysis. So results. So we only had 7 events to work with, so very small data set. But we just wanted to get like a brief understanding of what kind of results we might be expecting, and just get like an idea of what we can do once we start getting more results. So in terms of theta, beta, and gamma, we see there aren't that many correlations in terms of R and B. Like the p-score is high, all of them, so they're not significant. But we do see some correlation for alpha, which is like 0.43. So we thought, cool, there was some positive correlation between the alpha frequency and their shots. We didn't look too much into it for now because we are still in the middle of collecting data, but something that we can look for further into. And in terms of the topo maps, so the one on the left is what the athlete rated as his best shot, and the one on the right is what he rated as his worst shot. And we just basically did a relative band power for each of the 4 bands. And kind of the more yellow it is, it's the higher the activity, and the more blue it becomes, it's lower the activity. So we can see that they actually show relatively similar distributions, but there is less activity in all 4 band frequencies for his worst shot, so the one on the right. So that was pretty cool to see. In terms of deliverables, we're gonna be doing a complete GitHub repository. We're still in the works of like cleaning it up and making it presentable. And then we'll have like Jupyter notebooks for all of the codes and documentations, because there are multiple different, so like scatter plots and the maps. So we want to document the steps in the way to make it as seamless of a pipeline as possible. In terms of next steps, so the limitations, the major one is the fact that this is pilot data, it's not always high quality. We don't have much of it also to begin with. So this is constantly evolving when it comes to collecting data. And it's kind of like trial and error. And hopefully as we collect more and as we work with more, we can fine tune that process. In terms of future directions, we are trying to look into getting more athletes. Time window analysis, so like pre-shot versus post-shot, or if you do different sports, different like time windows. And the ultimate goal would be to kind of have like a machine learning model for real-time feedback, but that would require lots of data. So that's probably like something months away. Yeah, that would be pretty cool to do at the end. References. What's this?
Especially ones that are like maybe more high density and I don't know, like I'm thinking fast. You can't really do create like a patch. Or you could, it would be
So we Sam talked to some professionals. And apparently, this wouldn't be as applicable as in a team sport, rather than like a solo sport like golf or tennis. So in terms of other sports, we'd be looking more into like those solo sports. Other than that, though, not really sure. We haven't really crossed that bridge yet. Golf is easier just because it's easy to rate your shot. Like it's a swing, like Wii golf resort, like pretty chill. But yeah, I think we'll have to figure out different metrics as we try and look into more sports. Tennis, we got no idea. Yeah, it could be like maybe like target practice if that helps. Like a bow arrow kind of thing that I'm picturing. If not, it could be like worst comes to worst, go into subjective, but that wouldn't be as like reproducible in a sense. But yeah.
From the patient samples that you collected, have you categorized golf players into a beginner, advanced?
Oh, we only had one golf player. And it was a professional, but only had one. Yeah.
I see. How would you try to expand the data size?
We're just trying to recruit more people. For now, it's easier to do golf because Sam has knows a couple people in golf. So as they come into Toronto or Waterloo, you could just collect it. But other than that, it's mostly reaching out to companies or to teams and just asking them if they're cool with it.
Thank you.
Yeah.
I'm just curious. So as the athlete sweat, that's gonna change the impedance or that's gonna cause a lot of noise to your signal, right? Do you see your signal is getting worse and worse towards the end of the collection?
That's a good question. I don't think so, as far as I'm aware. I think from what I've been told, when she was collecting it was near the beginning of their training session, so it wasn't as labor intensive, might I say. But I would see that as an issue if you go to longer sessions. So that is something we should look into, yes.
Oh, I see. Also, they're just practicing for golfers, for example, they're just practicing. They're not actually on the court.
No, it's a simulation, so it's kind of you still have a club and a ball, but it's computer generated in terms of where your shot is gonna end. Does that make sense?
Oh, I see. I thought you were actually collecting data during the real games.
No, not during the games. Yeah, it's more during practice sessions.
I guess. Sorry. Just a drop off question with that. So I guess are you also controlling for same time of day and if it's the beginning of the training session or something like that where you're not as fatigued or performance is kind of consistent? Are you also controlling for time of day or other aspects that could affect?
Yeah, we try to do it at the beginning. In terms of time of day, it's hard because they also have their own stuff to do. Yeah. So we try to keep it as consistent as possible. So the most we can do is try to keep it at the beginning so that they're not fatigued. And then in terms of other time related variables, we try our best to keep it during the afternoon and stuff. But yeah.
Jumping off the bat, have you considered getting multiple time periods on the same person to sort of see the consistency? Yeah, or to take account for time differences within person.
Yes, we haven't gotten into it, but we have considered it. Sorry. Yep.
Yeah, thank you. It's very very innovative.
research, and I'm just wondering, are you planning to do it like a longitudinal one to see like how it goes? And I'm just wondering, how did you come with like buffer like in the first place? And what are some of the challenges that you go through?
In terms of how we came up with golfers, I don't know the answer to that. Sam was the one who started collecting it before BrainHack, I believe. So yeah, but in terms of challenges, I think that one of the biggest one is trying to get athletes to actually sign up for this. Because I know like some like coaches and stuff, they don't want their players or even their data to be out. So data is a huge like publicity of data is a huge thing that you have to look out for. In terms of longitudinal, I think once we can start showing results, it'll be easier to recruit more people, if that makes sense. But like that initial like jump to getting something that we can show off as a result does be the hardest thing for now.
Oh wait, okay. So is there any, or maybe I don't know if we looked at it, but is there any relationship between amount of noise and stroke quality? Like the amount of cleaning that needs to happen in stroke quality? Because like my main word would be like, maybe they do really well if they blink, and then the EEG is bad, but the stroke quality is great, you know? So, or like, yeah, or they're moving in a different direction.
Yeah, I see what you mean. In terms of that, to be honest, I'm not really sure.
Yeah.
I would imagine there would be
use like number of artifacts and
Yeah.
correlate that
Correlate that across trials to show that whether or not that's an issue.
Yeah, we could. I could try doing that. Good. Cool. Okay.
I think that was a fun presentation. A lot of people are getting antsy. I want to thank you all for sticking around so late. I guess we have some final, we're gonna send out the final feedback survey. But that's okay. I want to know